Once you are logged into the iLab operations software, you can request services from our core by clicking the Request Services tab. This will produce a list of services that you're able to order from our lab. To order a high-volume sequencing or plate sequencing, click this item here, and to the right, select Request Service. This will open up the service request form where you will tell us about your project. Please be sure to complete all required fields. The plate label is a user-defined field. This may be how you name your plate. In this grid, you'll provide us the names of your samples, the well location in the 96 well plate where we can find those samples, and the primer to be applied to each sample. You can enter these one at a time. And feel free to type in your own primer, or alternatively, you may select from our drop-down list. Since this is a 96 well plate format service, you may find it easier to paste this information from Excel. You can download this exact grid and fill it out in a list format, and if you have your information stored in a graphical plate map or a 96 well grid, you may find it easier to paste from our grid formatter. Download this file by clicking here. When you download that Excel file and open it on your computer, you'll be instructed to paste your sample grid and primer grid below in the spaces provided. You may navigate to where you have this stored in Excel or another program. Copy only the sample wells, not the coordinates themselves, and paste them where directed. There's our sample grid, and now we'll define which primers will apply to each sample based on their position in the plate. This Excel document has formatted my graphical plate maps of samples and primers into a list that corresponds with the expectation in iLab for the submission. Copy just the sample records, none of the headers, starting at row 24 and going through the end of your sample records. You can paste this in your submission here. When you scroll down, you can check and verify that your samples and your primers have pasted in an order as expected. Next, you're going to define the template type, the template length. You may have to average as this applies to the whole plate. Whether or not you want cleanup of these PCR products, and whether or not you want normalization. These are options. If you choose not to have us quantify and normalize your products, you're going to be asked for a concentration. Again, this applies to the whole plate, so you may have to come up with an average representation. These options here don't apply to every submission, and they probably don't apply to yours. Please contact our core staff if you are uncertain as to whether or not you need DMSO, max template volume, or a two-step reaction. If this template plate will be used in multiple submissions, perhaps with a different primer plate, click this button here. Finally, you'll be asked to direct how many plates are in this submission, and the default will be 1. Enter 1 and click Add Selected Services. Once you've completed those fields, click Save Completed Form. Finally, for external users or individuals whose funds are not located at the University of Arizona, you'll be directed in this section to provide payment information. If you have a PO number, you can indicate it here. If you do not have a PO with us and you will pay by credit card or check, simply enter that here. We'll contact you for payment details after the service is complete. Once you're done filling out this entire form, click Submit Request to Core. This will take you back to your dashboard. It's very important that you copy this number, the final digits of your service ID, and attach it to your plate submission, so that way we can recognize and identify your submission when we receive it. If you have questions about submitting to UAGC using iLab operations, please email our lab at uagc@email.arizona.edu. We look forward to working together.